Liverpool, who will be presenting to us Drosophila genetics, which is actually going to be the beginning, and I'm sure that we are going to benefit immensely from it. Wasting much time, I will have to hand over to Ahmed Badmos.
Right.
Thank you very much.
Right. Thank you very much for the brilliant introduction. And I am going to share my screen. I hope I'm sharing the right one. So yeah. So right. Thank you very much for the brilliant introduction, Doctor. And yeah. So yeah. My name is Ahmed, and today I'm going to introduce you guys to a bit of what we do in flies and what you guys are doing actually. So. And it's just because flies are fascinating, and during the course of this presentation, I'll be honest that I'm not gonna tell you guys about new stuff, but I'm gonna build on what other people have developed and what other people have done in the field, and those are the things I'm really gonna talk about. So also during the course of this presentation, you can stop me in between and be like, oh, you've got a particular question, and then you can ask. Also, what you can do is you can also make sure that you take notes of the questions that you want to ask, and also ask like later on, if that makes sense. And also, since this is going to be a upcoming meeting, this, like I said, this is going to be more of an introduction. And also, I want this to be more of a discussion rather than a lecture, because like I'm not the island of knowledge here, and definitely there are a lot of people that knows this or can teach this better than me. So I want this to really be more of an interactive class or more of an interactive discussion. So yeah, so I work with flies, and I'm very, very, very excited to always work with them, because also like they're just a source of motivation because of the cool things that goes on in the wild. So I really love working with flies. And so today I'm just gonna tell you a bit of the things that have been identified so far in flies, and the things that we can do. So without further ado, okay, I need to click on the screen and then move on. Yeah, so my talk is gonna be focused on a variety of subtopics, so including some feedback from the survey, the Drosophila genome. I'm going to talk about sex determination and some of the cool technology that we've got in flies. Also, I'm going to talk about how we can use flies as a system for discovery, and also I'm going to introduce you guys to genetics. Like I said, it's just going to be an introduction. So also, I'm going to introduce you to some online resources. And by this time, I think, because I'm very sure you've listened to several people, and I am optimistic that you also start looking at the online resources yourself and also trying to figure out how you can go about them. And what you can do with this type of discussion is you can begin to ask, oh, I'm trying to find this, can you help me? So I think I want you guys to get to that level as soon as possible. So from the survey, people want to learn about Drosophila genetics. People want to learn about glucose metabolism. So Drosophila anytime you want to teach genetics in a class or in a lecture, so Drosophila is the basis of genetics. So anyone that is going to teach genetics is going to mention Drosophila, because it is just a founding organism for genetics. And you can study glucose metabolism because they feed on glucose, they feed on sugar, and they've got all the genes that are conserved in the glycolysis pathway in the Krebs cycle. So you can begin to study that type of metabolism in them. And you just have to focus on what gene do I want to focus on. Do I want to focus on the whole organism? Do I want to focus on a particular gene? So you need to really nail what you want to study down. And you can do that with a cool technology and cool resource we've got in flies. Some people want to learn about how they can genetically modify flies. That is fantastic. Genetic modification, transgenesis, that is the bread and butter of fly genetics or in the fly world. Without that, there is nothing like fly experiment. So we can easily do that. And these are things that you're gonna learn over the course of this discussion. So also people want to learn about the fly physiology and the tissues in fly. So working with the whole organism is fun, is fantastic. but you want to go to the, let's say, cellular level, you want to go into the organelle level, you want to go into the tissue level. So you can also do this by dissecting flies. And like I said, the online resources involve some YouTube videos that can guide you on how to dissect them. And assuming we're doing this in real life, you know, we can easily do some dissection in the lab and show you how to do that. Also, people want to learn about nutrition. Similar to glucose metabolism, you can learn about lipid metabolism, you can learn about protein metabolism. You can learn about all these in fly. And also, the most important thing, how to do a successful crossing. That is really the key thing in fly genetics. So you really need to be able to understand your crosses. And once you get a hold of how to note the phenotype, how to cross things, you are good to go. Also, people want to learn about mutagenesis and microscopy. So mutagenesis, I would say, is an onset to fly genetics. So you need to do some mutation. You can start with mutation and trying to understand the mutation, or you can have a system and develop mutation that's going to affect that system. So these are things you can do in flies. Microscopy, fantastic for cell biology, for developmental biology, for stem cell researchers, for people that work on regeneration, people that work on disease. They depend on microscopy to really understand what is going on at the cellular level and at the tissue level as well. So you really need to understand a bit of microscopy, not detail of microscopy, because you're not developing a new microscope. You're only trying to image something. You're only trying to look at something. So basic understanding of microscopy is fantastic, and that will make your life really easy when you work with flies. Also, you want to understand how to tackle Drosophila research and you have to use Drosophila to tackle human disease. That is really the end game, isn't it? So like you want to, so the reason why we, as biochemists, as cell biologists, as physiologists, you want to work on thing or on a particular model that can allow you to understand what is really going on in human, in mammals, in us. Is that okay? So Drosophila is fantastic for that, and things you can do with that is you have to start with the genetics, and then you can build that up to human disease. So, and also people want to learn about the Drosophila gut. Fantastic. This is really specific, and I really like that because the Drosophila gut is really fantastic. And I've also got some illustration at the end that's going to show you the cool stuff that you can do with the Drosophila gut. So you can study a lot of things in the gut, and this is a cool model, fantastic system to study a lot of things, include disease, include regeneration, include metastasis, including infection, include a lot of things. And even there was a seminar online. So I don't know if you guys participate in that seminar that is like flying through the gut. So this is basically for researchers that work on the gut, and people have done a lot of cool stuff with that. So there are a lot of cool research going on in the field of Drosophila gut. Okay, sorry, I kept on admitting people, so I'm just gonna take a break and admit. Okay, good. So I'm going to start with the Drosophila genome. So the Drosophila genome was elucidated in 2000, and there are several people that came out in various journals about this genome. And what you can appreciate is Drosophila has got 4 chromosomes. These chromosomes, they occur in pair diploid. Is a diploid organism. And this chromosome X, 2, 3, 4. So these are the names. So the names, these are very important for when you're trying to identify the location of gene on the chromosome. So you want to identify, is this gene on the second chromosome? Is it on the 3rd chromosome? Is it on the X chromosome? These are the 3 major chromosomes that we use for genetic crosses at the minute. But people have started looking at the 4th chromosome, especially because they've got the chromatin on them as well. And you can begin to understand what are the genes that are required for things that we don't even know about. So people have started to study the chromosome 4 as well. And there are a lot of fun that the US is pumping into people that study the chromosome 4. So, but I'm gonna, for this part of our discussion, we're gonna focus on the X, the 2 and 3rd chromosome. And it is of interest that this chromosome have got a lot of cool stuff on them. They've got genes. You can balance them. You can use them for genetic crosses, and you can do a lot of things with them. You can manipulate them. You can look at location of genes, you can look at location of mutations. So, and this won't be possible without our ability to look at what is really the organization of the genome. And like I said, there are about 15,000 genes in Drosophila genome, so you can begin to tease apart these genes. And like you guys already know that all these genes, or most of them, that's 70% is like most of them, isn't it? So most of them are conserved in humans. So you can easily study things from Drosophila and you can extrapolate to human. So they are fantastic. So they've got this called genome organization. So, and you can read more in the reference below. And also, I'm going to tell you about how sex is determined in flies. So for you to have a male fly, there's something that goes on in them. For you to have a female fly, things goes on in them. So, and most of the things that goes on involve both transcription and splicing. So mostly splicing. So what happens is you need to splice, or you need to make sure you can express this gene that is known as double sex for female, and that really gives you your female characteristics. And also it's all only based on the dose of the X chromosome. So when you've got higher X chromosome, you can have female characteristics. When you've got lower sex chromosome, which I'm going to show in the next slide, then you've got male characteristics. And the reason I'm showing this is because there's some phenotypes. So when you work in the fly lab, you realize that there's something we call dimorphism, that you can see both male and female characteristics in some of the flies. So that is just because they fail to decide the expression pattern. So do they want to express double sex male, or do they want to express double sex female? And once this pathway is disrupted, then you can, so the cool thing you can do is you can begin to, so you can change a female fly into a male fly, and you can change a male fly to a female fly. So that's cool, isn't it? But you just have to understand what are at the genetic level, at the gene level, and understand what is really going on with the genes. Is that okay? And also understand how you can manipulate them easily. So, and what people have done, especially for the Drosophila god people, is you can make a male god in a female organism, and you can make a female god in a male organism. So this is just cool, and it is research, and you want to really understand what will happen if you make that, and what is going to go on. So that is just something that I said I should mention, and for us to really clarify. So moving on to the tools in flies. So one of the basic and one of the most important tool is a classic forward genetic screen. So, and it is the most ancient, like it's been around for years and for decades. So this is where researchers feed flies with mutagens. And what you can do is, because this is random, you don't really know what is going on in the organism. So it's random mutation. And then you try to map that mutation. So, and this is laborious. A lot of people do that. People have done this for, like, so when you want to look at genes that are required for development, you generate mutants in the male, you cross it to female, you look at the offspring that have got the mutation, and then you outcross them if the mutation is still there. So you can do several crosses, and you can do several generation of crosses, and you can begin to understand, okay, this is a particular gene, though we don't know the name, but this is a particular gene that is required for this, and then you can grow up the stock. So let's say, for example, you want to look at genes that are required for wing development. You can be like, okay, we've identified this gene, but we don't know the name, but we know that it causes this to the wing. So that is what the forward genetic screen is about. We're trying to develop something, we don't know the name, but we're just going to give it the name Notch because the wing looks Notch. So, we don't know the name because the eye looks white. We're just going to give it white. We don't know the name because the eye look red. We're just going to give it red. So that's just what forward genetic screen is all about. It's about, I develop a mutant screen. What was the phenotype? Okay, I'm going to give it that name. So, and it's also a game of number that, you can screen for thousands of genes, like let's say 1,000 genes, and then you can begin to realize that, okay, it's only, so you realize that 2 of them are the only one that are right, or 2 of them are the real thing that you're looking for, and discard the rest, or other people are going to follow up the rest. And this has won people Nobel Prize, and even you listen to Ross mentioning people that won Nobel Prize on development. So I'm not going to go into detail of that. So, and like I said, we've now moved on to sequencing these mutations, and people have now begun to name them, like giving them proper name, looked at how they are conserved, and also look at, so are we good? Good. So looked at how they conserved in human, and also looked at how the pathways that they fall in. So, and then begin to characterize what is upstream of them and what is downstream of them. And that is when you begin to look out for answers. So assuming you develop a mutation that causes the brain to rip off. So you want to look out for something that can rescue that. You want to look out for, assuming it rip off in half, you want to look out for something that's going to rip it off completely. So these are called either repressors or enhancers that you can then screen for. So that is what we call it backward genetic screen. So forward genetic screen is you're looking for that mutation that is going to cause your effect, and backward genetic screen is you're looking for something that is either going to rescue or is either going to add to the problem. So those are things that people have done, and with the facility that we've got now, sequencing, you can begin to sequence your mutation and really target that particular gene in that particular location of the genome. So also, for genetic screen has been replaced by RNAi or CRISPR screen. So these are just fantastic, and these are cool because unlike the forward genetic screen or the use of EMS, this, you don't really know what's going on in the system. But with RNAi, you can be like, okay, I want to screen for this family of protein. Okay, let's look at glucose metabolism, the genes that are required for glucose metabolism. Okay, I want to screen for all the genes that are required for glucose metabolism. And then what you then do is go onto the flies and then look out for the name of the genes and then screen for RNAi. And then you order the RNAi, so there's stock centers that sell them, and you order them, and then you screen for them. So you really know what you're screening for, but you don't know the answers that you're going to get, but at least you really know that, oh, this is the gene. So assuming you got an answer, you can tell easily that, oh, that's the gene that's causing the problem. The same thing goes for CRISPR, because these are targeted. You know the genes that they're targeting. It is so you can tell that, oh, this is what is really going on, and this is the particular gene that's causing that. So these are more cool, and they're just straightforward and easy to do. And I think behind this generation now. So for genetic screen, I don't know, I've never seen any paper recently looking at that, but you can see loads of CRISPR screen, loads of RNA screen. And I think that is what we should be really looking forward to do and add into our research portfolio. So yeah, so that's been replaced by that. And as I mentioned RNA already, but the key thing to the fly world, or the reason why flies are so cool, is because you can develop transgenic flies. So, and how can you develop transgenic flies? So transgenesis is basically you bringing in something that is from the environment or something that is different from the system into that system. And we can easily do this because flies can transform P elements. So P elements, there's basically plasmids. So plasmids are, they're in bacteria, and so there's a form of gene transfer. So we've got horizontal gene transfer, that you can transfer something from the environment, and plasmid is one of the things that bacteria can transfer to the environment so that other bacteria can incorporate. So the same thing goes for flies as well. So flies you can clone plasmid into flies, and then you can incorporate it into the system. And mostly they do this at the cellularization phase in the embryo stage. So you inject this plasmid into the system, and then immediately after cellularization, before the pulse form, they then inject the plasmid into the system, and you can look at what you're looking for easily. So you can easily do some proof of principle. Assuming you're doing transgenesis yourself in your lab, you can do some proof of principle to look at, okay, we've done this transgenic line, is it true or not? So you can there are things that you can clone for that's going to tell you that answer. So, but you don't really need to do that now. There are a lot of companies that can do transgenesis for you. Best Gene in Cambridge, they've got people that do that. Also, Manchester is trying to start this. So Andrews was mentioning that they're trying to up the game soon. So they're also trying to make sure that they can also do some transgenic flies in Manchester. So these are places that we can go to and we can order these flies and we can work and play with them. So so I can go, so because you need some molecular biology knowledge to do the transformation. So in the next lecture, hopefully I can mention some of the molecular biology tricks that we use to clone stuff into flies and generate transgenic flies. So, but I'm gonna mention some of the systems now. So one of the cool system and like the most widely used system and the most simplest system in fly world is the GAL4 system. So basically what this does is you've got a transcription factor known as GAL4 that is under the promoter of a particular tissue or cell. So you can use, so this goes to cell specific, that is how cool it is. And then you can begin to express a particular gene under your UAS construct. So what you do is you identify your gene from the database. So you also need to look at the database, look for the normal gene construct that, oh, okay, my gene is ACTGGGG. So you look out for that gene, and then you clone it into a plasmid that has got UAS tagged to it, or it can be tagged to other stuff as well. So, and what you can do with this cool stuff is you can do several things. One thing is you can knock down your gene. So using RNAi construct, and also you can express your gene. So let's say, for example, you've got a mutation and you're trying to ask, let's say you've got a mutation that kills the animal. And then you're like, okay, that is the mutation. But one thing you can ask is, can I rescue that lethality? Can I rescue that death by putting back this particular gene? So these are the cool stuff you can do. So, and because it is an old animal, what you then need to do with your GAL4 is you need to inject, or you need to make sure that you're driving with a particular promoter that is expressed everywhere. So people use actin GAL4 for that. People use tubulin GAL4. People use other GAL4. So you can use that to express things everywhere. And this just give you a flavor of what you can do with flies. And the system works like that. Oh, you've got two flies, cross them together, the offspring is going to generate what you're looking for. Easy, isn't it? And then what you then need to look at is, okay, so it can be a bit complicated, because then you can't just do anything. You then begin to look at what is your interest. So you need, so it's a top bottom thing. You need to understand, my interest is, okay, I want to look at the wing. What is the GAL4 that I need to look at? What is that gene that I want to look at? And then what is the readout? So your readout can either be phenotype, that, okay, I want green wings. Your readout can be, oh, I want to look at the molecular level. So you can break down the wing, and you can run it on, let's say, Western. You can run it on, let's say, SDS. You can do mass spec, or you can look at the protein level in the wing, and you can begin to compare. Okay, when we overexpress, when we reduce, these are the things that are really going on. So these are the things you can do with the GAL4 system. It's really fantastic and really cool system. So also, we've got the clonal analysis. This is mostly used for, you can use this for mutants, and also you can mostly use for mutant actually. So because what clones does, so what you need to do is you need to add what we'll call an FRT site. So FRT site is the flippase recognition target. And then you've got a flippase. So flippase is an enzyme that can flip out this recognition target. Is that okay? And so you've got a strain that carries that. And then you've got a strain that also carries the FRT and also your mutant of interest. So definitely you might already know this mutant, or you might have a guess that, okay, I think this mutant is on 3L chromosome or on 2L chromosome. Okay, if I add in FRT, let's say FRT 42D on this, what is it going to do with combination? So then you can guess, because you don't know. That's the power of science. And once you know, you don't need to guess. You really need to tell us the fact. So that is what the GAL4, that is what the FLP system does. So, and then you cross them together, and then you can begin to look at the patch of clone. So these are negatively marked clone, because what you've done is that your mutant is on. So your mutant is lacking GFP. But what has got GFP is the environment, is the background, or those are the control cell. And then you can begin to look at what is the size of my clone. are my clones dying? So you listen to the Pidini Labs lecture during the Drosophila Africa workshop. So she works on cell-to-cell competition, and these are the cool things that she does in the lab. So you can look at, okay, the size of my clone, is it because the environment is killing them? Are my clones killing the environment? So you can screen for genes that can do that. Also, you can look out for, let's say, the gods and be like, okay, we need to, so during regeneration of the gods, there needs to be proliferation. So can my mutant proliferate? Can they not? Why are they not proliferating? So you can begin to ask some intelligent and sensible questions. So yeah, and so that's just tell you. So this is slide just saying the same thing and how the principle works. So so the principle is that you need your FRT, cross it to a GFP. So your FRT that has got the mutant, cross it to a mutant, sorry, to a strain that is carrying GFP everywhere. The flippers is going to do the job. Then you're going to have what we call a recombination. So recombination is just where homologous chromosome pair and unpair as well. So then you can transfer some chunk of the GFP to your mutant. You can see that. So the GFP is here. You can transfer some of them there because of the flippase, and because that's done the recombination. And then you can have these two sister chromatids, and then during mitosis they tend to separate, and then you can you can develop two cells that are mutant for your cell type and something that is wild type for your cell type. Is that okay? So then you can begin to look at, do I want to look at cell death? Do I want to stay with the caspase? Do I want to? Then you can also separate these cell types. So so there's something we call facts that you can use to assort your cells, and you can separate them based on the fluorescence, and you can begin to look at the protein level. You can look at the transcript level, and you can do a lot of cool stuff with the system. So that is the FLP FRT system developed by the Rubin Lab years ago. So also we've got the MARCM technology, and if you are at the Drosophila Africa, I mentioned this during my talk. So what this system is similar to the FLP FRT, but this is more fantastic because then instead of you negatively marking your clone, you're positively marking it. So what that system does is you've got the same thing. You've got your mutant of interest combined with FRT. You've got all so we call this tester strain. So this has got heat shock flippase, so you need to heat shock that so that you can activate the enzyme. You've got the tubulin GAL4 that expresses GAL4 everywhere, and US GFP that is under the effect of the GAL4, and that should express everywhere as well. And you've got this cool thing that we call tubulin GAL80. So in the survey, I mentioned that if anyone knows something about Target, so Target is a system that was developed to inhibit the GAL4. So what that does is that, oh, you've got GAL80 in the system, GAL4 is not expressing. But as soon as you've lost your GAL80, then GAL4 is going to be expressed. then GAL4 is going to be expressed. So that is what Target does. But in the MARCM technology, what you do is so you've got that tester strain that you can then cross to these. So all these are basically doing some fly crosses. So you really need to understand your fly genetics or your crossing scheme properly. And then once you cross that, you generate offspring that is going to carry the tester strain, and also going to carry your mutant of interest, and also is going to carry the tubulin GAL80, because this is on a separate chromosome, and its chromosome is always the same chromosome as that, but different pair of chromosome, isn't it? So, but they should be on the same side of the chromosome. If that is on the 2L, your GAL80 should be on the 2L. If that is on the 3R, your GAL80 should be on the 3R. So then you generate that offspring, then you shock your offspring. So you shock this particular fly, and then wait for, so everything is going to do itself in the system. At the molecular level, cell division is going to occur, mitotic connection is going to occur, similar to the FLP FRT. And then you can begin to look at mutants that have got GFP, and then you can begin to also look at wild type that are lacking GFP. So, and then you can create, just similar to the FLP, create patches of clone. But it is cool because you're positively labeling your clones and you can see everything clearly. So, and like I said in target, any cell or cell types that are expressing the GAL80 will lack GFP because the GAL4 is being inhibited. So this is more of a biochemistry, cell biology, and molecular biology stuff. So right, so that is it. Those are the technology, and those are things. So we've got other technology in the flies, and there are also several technologies that I've not mentioned. So, but over the course of the discussion, we're going to mention some of them as well. Now, so I'm going to go into Drosophila as a system. So from Andrew Sprungkop, and he's done a fantastic drawing, and it's just so cool when he draws in, and you can develop some skill to do that. So what he has done here is he has compared the system in humans to Drosophila, and you can begin to appreciate that, oh my God, they've got similar stuff as us, and they've got everything that we've got. So, and that allows you to really want to understand and really want to study them, and once you study them, you begin to appreciate what you can really gain and learn from them. So all these are on the Drosophila for School website or Drosophila for Public website. But yeah, you can find these online. So assuming you want to use for lecture as well. So, but the gist of this slide is just that flies have got a lot of things in common to us, and they've got similar system to us. And then I'm gonna now mention a bit of things that we can do in flies and how we can use them. So, and then I'm gonna focus on the Drosophila gut. So what you can do is and then even looking at the gut, you can look at different parts of the gut, and you can look at the gut from the embryo to larvae to adult. And it is cool that you can not only you can look at the gut, but you can look at the part of the gut. So you can look at the hind gut, which is labeled there. You can look at the mid gut. You can look at the hind gut, that is the hind gut in adults, and that's the mid gut. So you can look at these things, and what you can appreciate, especially in the mid gut. So people use that to study regeneration. So, and in this mid gut, there are about 3 cell types in the mid gut. So they've got the stem cells, they've got the the enterocytes, and they've got the epithelial cells, and they've also got the stem cells that also give rise to the enterocyte cells. So enteroendocrine cells, I mean. So then you can begin to appreciate what is that particular gene that really switch, or that really allows this particular cell to be differentiated. Why do we have the epithelial cell, and why do we have the enteroendocrine cells, so the EE cells? So then you can begin to understand what is that particular gene that allows us to understand their fate. And if you listen to, I think, Julia Cordero, she also worked with the gut, and she mentioned some of the things that she's done in the lab, and that is just those are the cool things you can do at the molecular level in flies. And not only that, you can also begin to look at communication. So you can begin to look at how is the gut communicating with the ovaries, how is the gut communicating with the testes, what are the differences between the gut in the male and the female, why are male prone to infection compared to female. So you can begin to look at all these in Drosophila gut, in Drosophila gut, and you can begin to appreciate the things you can really do with the fly gut, and also systems you can work, other systems that you can work with in flies. And not only can you look at regeneration in the fly gut, you can look at infections. So you can begin to look at, okay, if I add this particular bacteria to the fly food, what's gonna happen to the gut? So is that gonna kill the gut? What's really gonna happen to them? And not only can you look at this in adults, you can also look at this in larvae. So like in Roskilde lab, what mostly they work with is they work with the larvae, they work with the adults, but they work with the hind gut. So you can begin to look at what are the tumors that you can develop in the hind gut. And that's in that area of research as well. So those are things you can do with the fly gut. And also you can look at development. So you can remove a particular gene, and that's going to prevent the gut from developing. So you need to ask, what are those genes? You can begin to screen for them. You can begin to look at them. So those are things you can do with the fly gut. So that's just looking at the fly gut as a proof of principle. But there are other tissues, there are other stuffs in the fly. So for example, people look at the eye disc. So people look at so we call this the eye antenna disc, and the eye antenna disc is linked to the brain. So that's the brain there, or the central nervous system, and the whole complex is known as the cephalic complex. So what you can do is you can develop a particular tumor model for the eye, and then you can begin to look at how are they invading into the brain or into the ventral nerve cord. Also, people have worked like immensely on the ring disc. So people have used this to also look at degeneration, to look at cell-to-cell competition, to look at cancer, to look at tumorigenesis, to look at development. So to look at cell death, to look at a lot of things. So you can to look at metabolism. So these are things you can ask. So for people that are interested in metabolism, let's say for example you've got a tumor model, and then you want to so you've generated the tumor model, let's say in the eye, and then the tumor model invade. So one of the studies on cancer is the Warburg effect, that they depend on on some glucose from the environment as well, and also they can convert the glucose to lactate, and then they can feed on that. So you can begin to screen for genes that is going to reduce that, or genes that are going to enhance that. So you can begin to look at those things in your tumor model, and you can begin to look at nutrition at the cellular and the molecular level, and you can begin to add that to, like, in disease. Okay, I want to look at in cancer, or I want to look at, or in the absence of cancer. So you can look at that easily in the larvae, because it is just easy to dissect, and you can really image them quite easily and carefully. And also in the adult, people have used the adult eye to study a lot of things. So because the adult eye is really small, so you can look at the roughness of the eye by screening for some genes, and people have done that a lot in the fly world. Also, you can look at the fly wing, so you can begin to look at, okay, if assuming we remove a particular gene in the larvae, what's going to happen to the fly wing? So you can to the adult fly wing, is it going to develop properly? Are they going to climb properly? Are they going to fly properly? So you can look at a lot of cool stuffs in this system, and people have done a lot of fantastic things with them. And not only doing that, but they can also, these things are also conserved in the human, and you can begin to use that to really identify the pathways and how to basically drug that particular pathway, and look at other effectors that can affect the pathways. So yes, so now, we're going to look at some of the cool stuff in flies. So I've mentioned a lot of cool stuffs, and the cool stuffs, they're just countless. There's just a lot of things that you can do with them. So one of the cool things is that the balancers, or the dominant markers. So we've got markers. So the wild type of, so if you look at Andres has got this on his website, and you can look at them in detail. So you can look at the wild type, so that's the color of the body, and you can look at the ebony color, and you can look at the yellow body. So you can begin to differentiate, okay, these are recessive, these are recessive. So if I've got them in the background, if I've got 2 of them, it's going to be ebony. So you can begin to appreciate what are the cool genetics things you can do with them. So also we've got for eye, so that's the body color. We've got for eye shape and eye color as well. So that is the white-eyed phenotype, because the eye is gonna be white. So, and that is the mini white gene, because it's got mostly people use this for like P elements, or when you want to transform the fly, so that you can know that the fly has got the transformers. So you label your plasmid with something that has got the white plus gene, and then it can do the trick. Also, we've got some balancers. So we've got the bar high, we've got the irregular facet. So people use this in the fly world a lot, and these are cool things that you can really do with the fly. So also, not only that, we've also got for wings. So we've got the curly wing, fantastic, fantastic marker. We've got the serrate wing. We've got the vestigial wing, which are really short wings. So you can do all these in the flies. And also we've got for the Bristol. So we've got the humoral Bristol, which is like tons of Bristol, because in the wild type you've only got two, but in humoral you've got tons. And also I really like this one, Stubble. So I work with that in the lab a lot. It's just a cool marker that you can use. And also for the Bristol, we've also got tufted. So tufted is just at the back, instead of having two or three, we've got like multiple at the back. So that's other markers. So there are also other markers that are not mentioned here. So, but these are things that once you've got, assume you've got a fly that you don't really know what marker it is, you can easily Google, and that's gonna give you the answer. So so what we're gonna just look at, I think this I'm at the end right now actually. But this is just a bit of an exercise for us to really think about, and then that's just gonna be our introduction into especially crossing schemes and how to really develop a genetic cross. So because that is really the key tool in the fly world, and once you really get a gist of the crossing, honestly, you're good to go. So I want you to really read this carefully. So you keep a fly stock that carries a homozygous lethal recessive for ataxin 7 mutant allele, and is wild type for the white locus on the first chromosome. So that is stock 1. However, for recombination with P elements, you need ataxin in a white background. So what do you think you're gonna do? So I want us to think about this for like 2 minutes, and because this is gonna be more of an interactive stuff. So and so sorry. So then I want us to really think about. So I want someone to really tell me, or if you can tell me what genotype of the stock you're going to generate, and then how you're going to do the crossing scheme. So there's no right or wrong answer. Honestly, I just want to have an idea of where to start from and how we can proceed with the genetic process. And like I said, I want this to be more of a discussion on rather than a lecture. So I think I've talked a lot. So I want us to discuss this a bit before the end of the meeting. So yeah, so I can leave the screen on, and if you think you've got the answer, so I'm just gonna stream the video. So yeah, so please, so I just want someone to tell us what is the genotype of the stock that we want. So read this carefully, and then we can say yes to your answer or no. So we need to agree on it. So do you want me to call someone then? Saying, so Doc, can I call someone? Am I allowed to do that?
Yes.
Okay, good then.
Yes, you can.
Okay, good then. Okay, because I can see some. So can Bolu tell us what genotype do you think you're gonna generate? If you don't know, that's okay, and we can move on honestly. Because I'm only calling the names that I can see. So please now, I'm not being political at all.
I think what we need is WW ataxin 7.
Okay, so is it so ataxin 7? So is it gonna be balanced or not?
Okay. Well, I'm not sure about that.
Very good, very good. Thank you very much, Bolu. That was a very good try, and I really respect that a lot. So because we want more than 1 or 2 answers.
I'm just waiting to wait for the
Yeah. Can Mary tell us what the genotype is gonna be?
Just waiting for the autoloader, which is the robotic arm, to be
Can Mary hear me?
by the luminal agents.
Yeah. So can you tell us what the genotype is gonna be, please? If it's not convenient, it's okay, no problem at all.
Okay. Since attacking mutant background, so the genotype would be ataxin 7 over WWIF.
Okay. So yeah, good. So yeah. Okay, thank you very much for that answer. So so just take note of what you said, and then we can come back to that. So can Yusuf tell us what the genotype is gonna be? So I'm gonna call one more person, and then we can move on. So Yusuf, not there. Okay, moving on. Can Zainab tell us what the genotype is gonna be? So then move on. Okay.
And antacin 7
Yeah.
over WWIF.
Okay. All right. Thank you very much, guys, and bless you for the answers and for the reply. I really appreciate that. That really makes sense a lot. So right, thank you very much, guys. So right. So the first thing you want to do when you're developing a cross is to look at the genotype carefully. So here, the first chromosome is this, the second chromosome is this, and that's the third chromosome for this particular stock. And for this second one, the first chromosome is this, the second chromosome is this, and the third chromosome is that. Is that okay? So the first chromosome here is wild type. It hasn't got anything on it. So that is why the eye color is red. Is that okay? Because it is wild type, it hasn't got anything on it. So ataxin 7 is a mutant. We cannot say it. So we just have to agree that, or we trust them, it is ataxin 7. But you can test that later if you want, because there are several things you can do to test that. So, but we can come back to that later. And then we've got the CYO. So, and that is the curly. So that gives you curly rings. Is that okay? And that is why you can see the ring is curly instead of straight. And the third chromosome hasn't got anything on it, so we don't care. That's okay. We are good. And then the second stalk has got white-eyed because it has got this W over W. So that tells you that, oh, this is a white-eyed fly, and because it is W plus and W plus. And this confuses a lot of people because when you see W plus, you're gonna be like, but it should be red, but it is not red. So W plus means it is gonna be like, it is, oh, sorry. So W plus means it should be white-eyed, if that makes sense. So WW means it is white-eyed fly. And if it is the irregular faceted that you saw earlier on. So if is what gives the fly this type of shape of eye. So you can see that is different from the first stalk. And curly is because it has got that curly wing. The third chromosome, we are happy, nothing is there. Is that okay? So the genotype that you want to generate, because you want to generate a genotype that is what? So it says a genotype that you can be combined with the P element so that ataxin 7 is going to be in a white background. So here, ataxin 7 is on the second chromosome. So the first thing you need to note is that my ataxin 7 must be on the second chromosome. And because it says ataxin 7 is what? It is it is lethal. It is homozygous lethal. So definitely you cannot have ataxin 7 all over ataxin 7. So slash ataxin 7, you can never have that because the organism is going to die. Is that okay? So you can only have it over a balancer. So then that introduces us to balancer. So what balancer does is to keep your stock moving. So to make sure that your strains are homozygous for those that are heterozygous. So sorry, to make sure that your strains are heterozygous for those that are homozygous lethal. Is that okay? For those that when you've got 2 copies of them, they're gonna die. So the balancers allow you to restrain that and to maintain them in heterozygosity. Is that okay? So you must make sure that your ataxin 7 is all over a balancer. Is that okay? And it can either be over a balancer that is this, because this is a balancer, or over Kelly, which is the same Kelly. So you need to, so that's the first thing. So ataxin 7 needs to be either over a balancer, which is if and Kelly. And the reason is because ataxin 7 is on the second, and these two are on the second. So it will always inherit something on the second. Does that make sense? And then you want to make sure that it is in a white background. The reason for now is that you want to make sure that your first chromosome has got that white over white. So you need to make sure that you need to think of a way that you can bring this white over white here. Is that okay? And just generate this. So white over white, ataxin 7 over Kelly or if. That is the stock that we want to generate. Is that okay? So you can take note of that. And then you want to design a cross. So when you're designing cross, I haven't got my pen. I don't think I can write on my screen. My iPad can do that. But yeah, so I wish I can write on my screen. So what you can then do is you want to then think of how can you set up the cross to make sure that in the inheritance you're tracking the inheritance and you develop the right strain that you want to develop, or you make sure that you've got the right stock that you want to generate. So the first thing you need to do is to look at, am I going to cross male and female, or am I going to cross female and male? So for this particular cross, because you want something on the first chromosome, the first thing you need to do is you need to cross this female of these guys to male of these guys. So take note of that. So I'm going to explain this in detail next week because I'm going to rush off soon. So yeah, hopefully next week. So yeah, so you need to make sure that, so cross this female of this to male of this. Is that okay? And then you then need to pick male flies. So you pick male flies that have got these, so W over Y. So you pick male flies that have got W over Y. And because you've crossed these as well, you can pick ataxin 7 over if. Does that make sense? So I'm going to explain in detail later. And the next thing you want to then do, so you pick male of that fly and cross it again to female. Because you've already got your W over Y, so you've already got white-eyed fly already because they're going to be white-eyed, then cross it to female. So any generation, any offspring that's going to come out from that is going to be either W over W or W over Y, because W is what is a limiting factor. Does that make sense? And then because you've got ataxin 7 over Eve, then you can pick for ataxin 7 over Kelly. So I am going to go into detail of these later, and also because it's going to be recorded, I'm going to want you to also provide me with the crossing scheme as well, and I'd love to see some answers from you guys, and then we can also move on into more complex scenarios and also more complex system. And it is just fascinating because this is very important for you to develop what you want to develop. So one thing is you want to combine this with P elements. What are P elements? P elements, you can use it, you can combine a lag Z to them, you can combine a GAL4 to them, you can combine GFP to it. So you can do a lot of things. So you can combine P elements that has got GFP into the promoter or into the as an answer to ataxin, and you can look at the localization of ataxin in the cell. Is that okay? Or in the whole animal. So you can be like, oh, is it expressed in the wing? Is it expressed everywhere? Is that okay? So also what you can do is you can look at GAL4. So you can clone a GAL4 into also as an answer trap to ataxin 7, and you can look at, oh, can we use ataxin 7 to drive a particular thing, to drive GFP, to drive LAC-Z, so you can do a lot of things with this simple genetic process. So they are simple, but you know, you really need to understand them, and those are really the bread and butter and things that you can spend years on them in the lab, and they just you know, just catch your attentions, and then you just really enjoy what you're doing. So those are the things that we do in the Fly Lab, and things that you guys really need to also do as soon as possible. So yeah, so I think I'm gonna end here. So I'm gonna end here now, and please, I want you guys to work on this, and then we can pick up from this in the next discussion. So I really appreciate those that answered the question, and it gives me a lot of joy to be part of this community and to really discuss with you guys today. So I'm gonna say thank you very much for listening. And if you've got, I'm sorry I haven't got time for questions today, but honestly, in the upcoming meetings, we're going to have more time to really discuss and really interact properly. So I'm so sorry about today. So thank you very much for the time. I really appreciate it.
Thank you very much.
Thank you, sir. Right, so I'm going to stop
really
sharing my.
You.
Right.
It's an exciting lecture. I would like to appreciate and thank Professor Oyeronke Odonola, the head of the Department of Biochemistry. Our mother in the department, we really appreciate her for joining us. Thank you, ma. I would like her to please and kindly say a word.
Thank you very much, Prof. It's really nice meeting you.
Okay, I don't, maybe she's not So by the grace of God, we are going to have another interaction with Dr. Badmos.
Ahmed.
And this is just the first of such. And by the grace of God, so we are going to send the link for us to be able to connect. In case you have questions, you can just drop them, and then I will pass it to him.
Yeah, please. Yeah.
Oh, okay, we are on with Professor Odonola.
Good morning again, everyone.
Why, good day.
Good morning, ma.
On behalf of the staff of the Department of Biochemistry,
right,
and especially the research team at the Drosophila laboratory, with special appreciation to the Dean of our faculty, Professor Olaleye, and to the immediate past head of the Biochemistry, Professor Farombi. Really like to appreciate Dr. Hamid for the wonderful this morning. It really brought down to basic genetics of Thank you very much. We also want
thank
to
you very much, Prof.
For initiating this series of lectures. We really need to build the team on the Droso research. Thank you very much, Dr. Abolaji. To all from the Department of Biochemistry, and I can see also so many students and the faculty members joining from other universities. We are really, really very good. Thank you all once again. Thank you for joining.
Thank you very much, ma'am.
Thank you, Prof. Thank you, Prof.
On the 4th of December, we'll be having Dr. Dara Biochemistry Department, University of Oxford, who is going to give us another presentation on SM fish. We're going to send the link to you. And also when we confirm the dates when Ahmed is going to interact with us, we will also pass this across to you.
No problem.
Before we close finally, I don't know if anybody has any comments before we go. Okay, on this note, we want to appreciate Ahmed once again. We are grateful for your time.
Right. Thank you very much, sir. I really appreciate being part of this. Thank you. Right. So it's very nice meeting everyone, and I'm gonna see you soon, hopefully. Right. Have a good day and enjoy the week ahead. Right. Bye now.
Bye.
Right. Bye.